And Buzz is of course also on the paper, and Dow I think you've also met in the system biology lectures. But what I like about this paper is that they actually use let's say bioinformatics methods to gain some understanding for like a more like a system biology topic. So I think it's a nice bridge between the two fields. Okay, so in this paper they look at basically we are trying to get any functional information of microbial communities, and they look at two data sets. But here we're going to focus on kefir. And in general, so what kind of questions can we ask from getting a metagenomics data set of a microbial community? Metagenomics is like a fancy term, but just means that basically get a data set or form genomics with multiple species involved. And usually we want to know who is there, so what the species is actually part of the community. And we want to know how many of each, so we want to understand, okay, what are the sort of amounts of all the species that are in there, what are they actually doing, do they perform any task, or, and then also, of course, really important, are they interacting? And that's usually the most interesting we want to get at, but also the most, let's say, hardest to understand. But we'll get to that later. So first, what is actually kefir? Some of you might actually have had that before. It's like a fermented milk drink. I think it's kind of hip right now. And it's a community of many bacteria and yeast species. And it has a low lactose concentration. Also, people who are lactose intolerant might be good. And it's also a so-called probiotic. But I put that also in quotation marks because there's a lot of debate that actually these probiotics actually do anything good for you of any of the living species actually reach your intestine, because they might just get killed in the stomach. So, but of course, people that sell these kinds of products would like to tell you that it's really good for you. But it's still, nonetheless, a very interesting microbial community. It has been around forever, so they can isolate some of these kefir, say communities that have been around really for hundreds of years. People really, so you can also do that at home. You just have these little round pellets, basically, let's just call them. And you can just always take a bit, put it in milk, it will ferment, and you can always just continue that. And so they're really well adapted to their to that environment to ferment milk, basically. And there's another very cool paper. They also looked at the structure of this. They did some very cool electron microscopy images. So we can actually zoom into that a bit. So they appear like these little balls, basically. If you zoom in, you can get a closer picture. And if we zoom in even closer, this is what one of these small little balls would look like. And if we zoom in a little more, this is now the electron microscopy images, we can actually see that there's a lot of these bugs tightly packed together. And if we zoom in even more, we get a closer picture. And so does anybody have an idea in these pictures what could be bacteria and what could be yeast? Yeah, what's your idea? Exactly, so these rod-shaped ones usually what we interpret as bacteria, and then the big ones is the yeast. And there's also already a cool picture to see that there's actually quite a big size difference between these. So let's say a like diameter of these rod-shaped bacteria we think to be about 1 micrometer, and then a diameter of the yeast cell can be like 5 to 10 micrometers, so they're actually quite big. But this is actually a very interesting microbial community, because usually what we study, it's just the soup, it's just things swimming around. But in kefir, they actually form these structures, and we also think that is also important for how they perform these fermentations and what order it is performed. And so that, it's very interesting to study it, but also very difficult, because there's many different species in it. And but to zoom in exactly, so in what consists of this community, so also in this paper, they actually identified more than 30 different species, and there's different stages of fermentation. So the main sugar that is available in milk is lactose, and that is fermented to lactate, and that's mostly done by lactic acid bacteria. And that actually lowers the pH, so at some point the lactic acid bacteria are not as happy anymore, they can't perform their tasks well anymore. But then we also have the yeast that are actually more resistant to lower pH, so they can still keep fermenting, and that also explains the low lactose concentration. But these metabolic interactions between these species are not understood. So is there any kind of cross-feeding? Is there one compound that can only be broken down by one species, and then the product is used by another one? That is really not well understood. And this can also be very difficult to understand. So here's also a figure from a very nice review, and this just shows the possible interactions of two species, right? So it's really just that we have in these boxes, there's one of the species, and the interaction can be positive, negative, neutral, and then in red and green we have the two species. And you can already see this is really complex, right? So we can have some sort of mutualism where they really benefit each other. That would be like a pretty symbiotic relationship. There can be competition, which I think also occurs a lot. And yeah, there can be parasites and all kinds of interactions. So you can really just see that, you know, just looking at it, being, oh yeah, I don't know, I let them grow together and I measure what is being consumed and what is being produced. It's really hard to understand. And yeah, if we have any more than two species, I think using genomic methods is really the only thing that we can do. And that's why I also use that approach in that paper. So more details on the paper, they actually have these 30 species. And the main question is that they want to understand, can they identify functional groups, right? So out of these 30, it's likely to assume that they might perform similar tasks. And they also want to see if we can get these interactions. And before we go into the data, we need a small primer of KEGG. Has anybody of you heard of KEGG before? Yeah. So it's also a very nice database. Most people use it for pathways. It also has more information. But so the abbreviation is actually Kyoto Encyclopedia of Genes and Genomes. Also didn't know that before yesterday. Yeah, it really contains all the relevant pathways and annotated genes. And also a term that is important to understand this paper is a KEGG ortholog. That actually just means that there are genes in different organisms with similar functions that are then part of these pathways. Does anybody know the difference between homologs and orthologs? Because we usually discuss homologs here, right? But what are orthologs? No idea?
Orthologs were with a shared ancestry.
Also homologs are really shared ancestry, exactly, and then orthologs can be the similar function that evolved, you know, but the ancestry can be different actually, but they evolved to have the similar function. So, and in that case, we don't care that much about the ancestry. We don't want to understand, you know, do they have the similar function because they have a common ancestor, but we really just want to know what is the similar function in a pathway. So they really care more about these orthologs. And here's an example for glycolysis in yeast, so one of the most well-studied metabolic pathways. And this is what the picture would look like in KEGG. So we'd have the pathway, starts up here with glucose, all kinds of different steps, and then you end up at pyruvate. And the, let's say, boxes that you see here would all be enzymes, so, and also these numbers would be ECs, so sort of an enzyme identifier. And then all the green ones are the ones that we do find in yeast. And this is so how you can imagine basically that they, how we use this database, and we actually just see, okay, in this pathway how many genes are present. And and this is not species specific, right? This pathway exists in most species actually, and we zoom in like a specific enzyme, like for example here I think would be EFK, and then that would actually be so you can see that yeast actually has two different kinds of PFKs, but then this is what we're interested in, right? So does this organism have the enzyme that can fulfill that function? Okay, so let's look a bit closer at the workflow that they use in that paper. So they start off with genomics, and they actually put that in brackets because the example of kefir is actually, they also use isolated species, so it's not proper metagenomics, but they still have genomic information of multiple organisms. And then they annotate this. That's really what we saw before. So they use, I think here they use BLAST Koala, which is a tool that also uses BLAST, and but also information that can be found in KEGG to identify genes that are an ortholog and can actually fulfill that function. For example, you would have different species, like we saw before. So one species can actually have two genes that fulfill similar function, and then in a different species there would be another gene, but that has also the similar function. And then if we have that, they perform a clustering step, and they can do that based on different information. So they can do that on the presence or absence of these pairs. So they really just can imagine that they have a table. Let's see. You would have the different species, and then 2, and then they really just see, okay, is this present or not present? This is really kind of a basic, let's say, algorithm, but that's also, like, the simplicity is quite nice of it, right? It also makes it easy to understand. So they really just see, okay, how similar are these species in the sort of genes that, or enzymes that we find. But another step they can also do, you can also do pathway enrichment, so that you actually see, okay, how many genes of a certain pathway does my organism have, and then the clustering happens on that, based on that information. So not only on the single KOs, but actually on pathways. And then they can identify functional groups. So, for example, let's say, I don't know, you have certain bugs that are have the green function, some have the purple function, and then from that clustering, they actually want to identify features. So features we had before in Sun's lecture today, but also the things that actually distinguish the functional groups. And yeah, and then if that works, they can actually make some kind of prediction about the interaction. The clustering algorithm that they use is affinity propagation. I'm also not an expert in that, so if you want to know that, probably get in touch with Bas or Ratz, but it's also not really important to understand the content of that paper. And then this would be an outcome. So after they did the clustering just based on the absence and presence of these KOs, they can actually have these 30 species and identify 8 clusters. So we can see that here on top, basically different colors would be the clusters it identifies. And you can also, you know, these kinds of things also make sense. For example, if you look at the species names, and then yeah, it makes sense that the species of a, let's say, similar, what level is that? And family, that they actually belong together. So that's nice. And so they are actually able to identify sort of different functional clusters. And they can do the same approach where they do the clustering based on pathways. And the result is actually quite similar. I mean, the image looks different, but you can also see that they also identify 8 clusters. What's interesting, there's this one bug that stands out. It actually is sort of in its own cluster. We'll get to that later. So that seems to be quite different from the other ones. But in general, it's also a nice outcome, let's say, so that you can just use that easy information of the KOs in order to find nice clustering. You don't need to do the pathway enrichment. But nevertheless, you do the pathway enrichment in order to get some more understanding and to actually see, okay, what is different between these functional groups, because that's what we want to get at. So, get these functional clusters. Similar results for the basically 2 clustering approaches. And if we then actually look at the pathway level, we can look at differences. So within one of these clusters, right? So we can just pick out, let's say, for example, here we can think what we do in examples. We pick out some of the lactic acid bacteria and see how they compare to each other. And then we can also compare between clusters. So we can take one representative of each cluster and then compare them to each other. So this is what it looks like within one cluster. So we have all the different species. This is actually also the one that was standing out a bit. And then what we see here is then in the basically rows, we have the pathways, how we would find them in KEC. And then in the colors, just the coverage of that pathway, right? So how many enzymes of that pathway are present. And here you see the scales. For example, black would mean that in that one bacterium here, there's a high coverage of alanine metabolism. But you can also see that it seems that, say, these are quite close to each other, and then this one stands out. And this already, say, gives some kind of possible application of this data, right? So imagine you were you work at some biotech company, you want to create a synthetic kefir community, and you know that you need this functional cluster, right, of bacteria in order to fulfill certain function. So what do you think? How many species do we need to, let's say, fulfill all the functions that are important for this functional group? Ten. Well, that's more than are here, right? So I think it's less than 10. Because there are only, 6 in that functional group, right? Any other guesses? Yeah.
I think 2.
Yeah, exactly. exactly. That's also what I would think. Yeah. So really, we have this one, right, which really has, for example, a higher coverage of this PTS system, whereas the other ones don't. So I think that's really important to have. But the other ones seem to have a, let's say, similar coverage of pathways, right? So it's like if we were to recreate that and we had to pick only two, I think we could just pick the one with the really long name and some of the other kefir species, basically. Exactly. So that's the kind of application. Huh? Let's check again. 152, so that's this one. And we need it for. Let me see. Oh yeah, that's true. Yeah, exactly. Yeah, yeah. No, you're right. Yeah, exactly. Yeah, yeah. Exactly. So if we were to pick that, probably the 152 and the 322 would be good choices actually. Yeah. Yeah, but this was say within one functional group, and then we can also compare the functional group to each other. So same kind of picture. We have the different pathways. They actually, so obviously there's more pathways, right? But for this picture, they, let's say, pick the pathways that are most different from each other. So it actually have the largest standard deviation between species, and also the same color. Exactly, and so we can see it, yeah, obviously there's way more differences between these representatives of the functional groups, and we can also identify a couple also interesting things. So for example, there's this one bug, or one that only has this high representation for flagella assembly and bacterial chemotaxis. So what do you think, what can we learn about this specific bacterium, about its location in the microbial community, or about what it's potentially doing? Yeah.
It's probably moving towards some type of gradient.
Exactly. So it's really one of the few that actually does have the required genes for making a flagella, right, and also for chemotaxis. So this is probably just sort of swimming around in the middle, right? It might not be part of that, it's a fixed structure. Exactly, very good. So this is also the kind of things that we can then learn from it, right? So it's really not just looking at one bug and seeing what kind of genes are there, but also comparing it to the other ones. And I think that can really give us the information. Yeah. Okay, yeah, yeah. If you haven't heard of that, so let's imagine we have some rod-shaped bacterium, and then a flagella would just be, let's say, something that comes out of one of the rod ends a bit, and then it makes a little turn like that. And then there's actually, compared to the length of the bacterium, there's a very long, let's say, tail. And just by twisting that around, they can actually, just move around in, let's say, their, watery environment. And so that's really a good sign that there is some motility of that bacterium. Okay, so and then also what's also very important. So when you study any kind of scientific publications, it's important for you to have a look. So what are the potential pitfalls, right? So where did it go wrong? What are issues with the interpretation of that? And one thing I found that what they also mentioned is that only 50% of the genes that they sequence could be mapped to KOs, right? So that could mean that you just miss certain pathways or you miss certain functions. So that's something to be aware of. And also what is also a problem for these microbial communities that they actually only looked at species that were able to isolate. So they only looked at the ones that can actually get from the community, grow by itself. And a lot of times in these microbial communities, you just cannot do that. It's actually really hard to isolate those. So it'd be better off to just look at the entire, let's say, sequence the whole system as it is. But of course, then you encounter other issues. But that's something to be aware of from this paper. So the conclusions that they, let's say, this method that they present here, this KO annotation and clustering, is really able to identify functional groups within that microbial community. Then the pathway enrichment really gives us clues about what is different between the functional groups. And there's also they could actually identify some interactions that could imply cross-reading. So if you remember that there's certain bugs that cannot make an amino acid, some can make that. So we could think that they actually exchange that type of amino acid. And now what do you think? How could we improve this analysis? So I really mentioned one aspect, right, that they just sequence the isolated species, but do you have any other ideas of how to improve that? So now we only have genomic data. Do you have any clues of what other data types we could use or other types of experiments we could do to get more understanding about that? Exactly, yeah. No, that's a good point. So, and I think also in order to validate some of the findings, I didn't mention that specifically, but they also did a different approach where they then try to reconstruct genome scale models, so which is just a, let's say, an entire metabolic network of all the possible genes in a organism from the genomes that they found in order to also improve the, let's say, KO coverage that they have. But indeed. But for example, do you think only because a gene is present in the genome that the organism actually uses it?
Yeah, so gene expression data.
Exactly, yeah. So I think that actually gives you a lot more information about what is really going on, right? So you see the genome is just the potential, right? Like, let's say all the genes that could eventually be expressed, but then if you look at the transcriptomics data, you can actually get a lot more information what proteins are actually there, what are actually being expressed. Okay, so that's a wrap about that paper, and just in the end, just want to mention that Fats, the first author, is also looking for interns. Might be a bit early for you guys, I'm not sure if you're all first-year students, but actually there's some very interesting projects. So in this latest project, they want to study the interaction of humans and lactic acid bacteria in their, let's say, co-evolution. So that's a very cool collection of lactose, just lactic acid bacterium strains. And they actually want to compare that to mammalian pathogens and to identify, let's say, relationships between mammals and microbes. And then they also want to be able to establish some kind of timing so that certain events that happened in the evolution of humans and lactic acid bacteria together. So very interesting research. So if you like this kind of stuff, really applying bioinformatics to, let's say, gain more biological understanding, this would be a cool project, I think. Okay, I think that's it for today. So then good luck with the programming exam. Yeah.
So which figure do we need to know?
Yeah, exactly. So I think the most important one for this is figure 3, which has these two, let's say, plots. So the two heat maps. It's in figure 3. We really want to understand, okay, how can we, say, from the coverage of these pathways within one functional cluster or between clusters, what can we learn about the community? Yeah.
One of the first figures you have different colors, but what color you're pursuing? Not in this one.
Oh yeah, just the heat map. Yeah, this is just say how similar species are to each other, right? So I think it's the colors that this is clearer. Let's say white is very similar, right? And then on the, let's say, columns and rows, you just have the two. And you have all the species, right? So you can imagine that you have here the list of species, you would have the same list of species here. So that means in the diagonal, it's just with themselves, right? So then they're obviously very similar to each other. And then I think the gradient is then going to, let's say, orange, and then green is very different from each other. And you can actually, for example, see here, right, this is very like big white box. But that means there's a lot of these lactic acid bacteria that are very similar to each other. Yeah. Yeah. Okay, yeah, thanks.
We don't have the computer class anymore. Yeah, it's quite hard, but I would like to know what my score was in the first exam, so I know how much I studied. Because if I scored like 40, I definitely have to